back to Science in Real Life. In part 1 of this episode, we used PCR to isolate and amplify a specific region from the genome of 8 species of the carrot family, and then we sent the PCR product to the magical land of the DNA sequencing facility. You're probably left wondering, why did they do all of that? Well, you're about to find out. Hint, it has to do with molecular systematics, which is just a fancy way to say that we'll be using DNA sequences to understand the tree of life. The scientific tree of life, that is, not the Terrence Malick movie, which I don't think anyone really understands.
How does molecular systematics add to more traditional forms of classifying species?
Well, traditional approaches to classifying species were based mostly on looking at the shapes, the sizes, colors, numbers of parts. That's what we call morphology, and also the way cells are put together, which is what we call anatomy. These systems have been really very successful. I'd say about 80 to 90% of the traditional classifications have been confirmed by molecular systematics. The 10 to 20% of cases where molecular systematics disagrees with the traditional systems are usually cases where there was convergent evolution. And that's when species that are unrelated and don't share a common ancestry might have evolved the same kinds of adaptations in response to similar kinds of environmental pressures.
So, for example, species that are all adapting to a dry, harsh desert environment might all have succulent leaves and spines, but they don't share a common ancestor.
Exactly. So making a phylogenetic tree is a little bit like making a genealogy or a family tree. In reconstructing family trees, we can go to our parents and grandparents and ask them questions, and we can also go to things like county courthouses and look up records. Well, we can't do that for species of plants because they've been around for millions of years, so there's no courthouses that have their records. So instead, what we can do is go back into the genome and look, because the genes have secrets about the way that the plants originated, how they're related, and the timing of those things.
Before we jump into data analysis, here's a quick reminder of the DNA region we isolated and amplified with PCR.
We're going to be amplifying a spacer region of DNA between 2 genes from the chloroplast genome. And the reason we're doing that is that the species that we're looking at today are very closely related. Their genomes are going to be very similar, and we need to find a region that's accumulating mutations much more rapidly than genes would.
So if I were building a phylogeny of distantly related plant groups, I could use a gene that accumulated mutations more slowly because there'd still be enough characters.
Absolutely.
Okay, so our PCR product, which contains millions of copies of this chloroplast spacer region, is analyzed by machines at the sequencing facility that can detect the order of the nucleotide base pairs in the molecules of DNA. This is the DNA sequence, which we can read like a sentence and use in our data analysis.
What we see here are 2 different strands. One's a forward and one's a reverse, so they're both complementary strands of DNA. And if we zoom in, we can see the actual base pairs, and those base pairs are based on these chromatogram peaks. All right, these are called chromatograms because the different peaks are of different colors, and the different colors represent the 4 nucleotides of DNA: guanine, adenine, thymine, and cytosine. And what we're going to be looking at here is to see if there are any differences between this sequence from one species and other species that we're going to be comparing it to. So those differences across species are what we call mutations, and those mutations are the way we build our phylogenetic trees. So the next step is to take the one species that we had and to compare it to all the other species that we're doing the same thing for. We have rows, and the rows represent the different species, and then we have columns, and the columns represent different individual base pairs that make up the gene across all those different species. So the colors are the same as we saw in the chromatograms, the 4 different base pairs, and then differences in the colors represent the kinds of mutations that we were talking about before. So there can be point mutations, and those are where there's just a little bit of difference between whether it's, for example, a cytosine here in the first 2 or adenines in the next 5. And we can also have length mutations, and that's where the sequences are of different lengths across the entire gene, and we have to line those up. So what we do is we insert gaps, but we've pushed over the sequence so that it lines up with the sequences of the other species that have the extra base pair.
Okay, so this group of species is missing that base pair, while this group has it.
Exactly. And so that sort of represents kind of a natural grouping of species that you can see there.
Okay, so how do we get from that alignment to this tree?
We take the sequences that were in the alignment and we analyze them with a computer program that looks for shared similarities or shared characters among these different species. And that computer program then helps generate this phylogenetic tree. So what we're seeing here is that the Belisi group represents a single evolutionary lineage. And what that means is that they all share a single common ancestor. All the Belisi groups would share a series of characters that can be mapped out along that branch. And another thing that this tree shows is that all the species that were originally placed in Bolesia come out together. This is a case where our sort of modern techniques are completely agreeing with and confirming the results of earlier scientific work. This is a tree of related groups in the same family, and what we see here is the taxonomy didn't fare as well. So what we're seeing now is, for example, this yellow group is the genus Astericium. And so traditionally we thought that was one genus. So what we would expect is that all the yellow things would come out together. There'd be a single common ancestor for those. But instead what we're seeing, for example, is that some species of Astericium in yellow and the green ones in Pozoa share a more recent common ancestor than other species of Astericium in the same group. So this is showing where our evolutionary trees differ from the traditional taxonomy, and it gives us a good example of the way that we are able to improve upon earlier scientific work.
It's just really amazing to me that we can go from PCR reactions to understanding the tree of life in a few steps.
Absolutely. PCR has totally revolutionized biology. We know that it's really important for things like the Human Genome Project. You've seen how it's involved in evolutionary biology and reconstructing these evolutionary trees, and also to give you a sense of the research we do here on plant biodiversity at the New York Botanical Garden.
Well, thank you so much, Greg, for joining us for today's episode. Thanks so much for watching Science in Real Life, and we'll see you next time.